tools that are specific to neurophysiology and psychophysics contexts that'd be helpful for my research, and actually be forced to learn Git and GitHub. And I'm happy to say that I feel like I achieved these goals throughout my time in the program, and I'll present to you the results from this work. So one of the main questions I ask in my research is, what sounds do animals hear? And the way we try to answer this question is by conducting hearing tests. But how do you test hearing in animals? Well, in the human animal, we ask them to raise their hand when they hear the beep, and you can do similar tests with other animals as well. Like here, this dolphin has been trained to nudge its snout on this ball hanging on the side of a dock whenever it detects a sound. Another measure of hearing that doesn't require training in an animal to respond behaviorally is to record its brain responses to a sound. In this kind of test, we present a sound over a speaker, measure the brain response using electrodes placed on the animal's head, and connect the electrodes to a computer to measure the response. And this specific type of test is called the Auditory Evoked Potential test. To know the range of sounds these animals can hear, we present sounds at different frequencies and amplitudes. So when I'm changing the frequency of the sound, it is like I am pressing keys up and down the piano. Okay. And humans perceive this change in frequencies as changes in pitch. And when I'm changing the sound amplitude, it is like I am changing how hard I press on a single key. Okay. And we perceive these changes in sound amplitude as changes in loudness. And thinking of sound at a more basic physics level, if I were to track the trajectory of this red air molecule, yeah, it's visible here, which is moving due to a sound wave passing through. How many times this molecule moves back and forth in 1 second is referred to as its frequency. And how far this molecule moves relative to a baseline position is referred to as its amplitude. And by testing the hearing sensitivity of an animal at different sound frequencies and amplitudes, we can create these curves called an audiogram, which shows the auditory threshold or the softest audible sound at a given sound frequency for this animal. So here, the audiograms of a human and a long-legged bat are plotted. And as you can see, the bat is sensitive to higher frequency sounds than humans, while there's some overlap. And these sounds that are above the human's audible frequency range are called ultrasound, and bats echolocate at these frequencies. And sounds that are below the audible hearing range of humans is called infrasound. And as you can see, these delineations of these physical frequency ranges are human-centric. Some examples of the utility of measuring auditory thresholds is that we can make informed predictions of how sounds generated by human activity can impact wildlife. So for instance, it has been shown in humpback whales that they can hear sounds from 80 hertz to 22 kilohertz or 22,000 hertz. And so we can predict that it can hear fairy noise passing by, which has sound from 1,000 hertz and below in the water as it's passing by. And on a more basic science level, I currently study hearing in fishes, and you may be surprised to know that fishes can hear, but actually all four-limbed vertebrates, including birds, mammals, amphibians, and reptiles, have ears that have evolved from fish. So humans, we get our ears from fish. And thus, by studying the auditory systems of fishes, we can learn fundamental processes in the evolutionary trajectory of the vertebrate auditory system, which can have wide-ranging applications. And so my current work focuses on the software and data analysis component of the Auditory Evoked Potential test. And through my collaboration with Dr. Rokem and my PI, Andrew Brown, I have built a MATLAB-based app that performs data acquisition and online analysis. The two main goals of this package is measuring auditory thresholds faster and more accurately than current standard practices in my field. So the value of doing this test faster is that it makes it possible to test in a greater variety of species. The auditory evoked potential test can still take like 1 to 2 hours to complete, And for species that are sensitive to experimental handling, it is unlikely that they can respond to sounds consistently during that time. And the motivation for improving the accuracy is that in our field, it is common for experimenters to determine if a response, brain response, auditory response is present or not just by looking at the brain waves. So to demonstrate our concern with this, I'll show an example. So this plot to the right is showing the frequency components of a brain response here. And higher values on the Y means more energy at that specific frequency. And so here we see a strong peak at the expected auditory response frequency band at the stimulus amplitude. So it'd be reasonable to say that the subject detected the sound. And as we decrease the stimulus strength, the peak of the auditory response also decreases to the point where it's hard to distinguish it from the noise floor in the signal. And the experimenter's job is to determine what was the lowest amplitude sound that still evoked an auditory response. And this is a very hard task to do just by looking. And so this method likely leads to inaccurate estimates of the auditory threshold. And actually, this last response at the bottom is a recording from a dead fish. It's common practice to do a whole set of tests on a dead fish as a control. And you can still see small peaks in the response, and that likely is representing electromagnetic noise that still gets in. So here is our proposed solution. In the ADAPT AEP workflow, the program will first present a sound over a speaker and measure the response from the electrodes. And then the signal from the electrodes will be filtered in order to improve signal quality. And then these signals are put through a response detection algorithm, and I'll go into detail about it next, where instead of an experimenter eyeballing the auditory response, statistical analysis is conducted to see if the response is statistically significantly greater than the background noise in the signal. Depending on the result of this step, the program will either go back to the first step in the workflow, present another set of trials, or if there is a response detected, it will progress to the next step. The program can also progress if the max trial limit that's set by the experimenter in the beginning of the program is met as well. Then the program will take all available data and fit a model to the auditory response magnitude data plotted as a function of stimulus amplitude. And I'll go into more detail about this later too. And one of the parameters in this model is the auditory threshold value. After the attempt to model the data, the user has a choice to test out a new stimulus amplitude or test out a new stimulus frequency or end the experiment. Okay, so going more in depth here, the response detection algorithm calculates if an auditory response is statistically significantly higher than the background noise. And it does this by comparing the electrode signals when the sound is off versus when it is on a trial level. And it specifically calculates the energy at the expected auditory response frequency bin. So we're in FFT land now, and compares these amplitudes. And this comparison of the auditory response to a relevant baseline on a per trial basis is especially helpful when you have non-auditory signals contaminating your response, like 60 cycle noise coming out of the outlets. And to compare these amplitudes at these two time periods, I use a statistical method called bootstrapping. And bootstrapping is a way to resample your data set to estimate the distribution of a measure of interest, like the mean. So you do this by randomly resampling your original data set with replacement, meaning that you can include the same data point more than once in your resampled set. And then for each subset, you calculate a statistical measure of interest, like the mean here, and then you create a distribution of these mean values then that you can conduct confidence interval tests on. And this doesn't require that your original data set or data to be normally distributed, which was nice. And the program specifically implements bootstrapping by first subtracting the electrode signal frequency spectrum of the stimulus off period from the on period on a trial by trial basis. And this trial by trial level subtraction is another method of improving the signal to noise ratio or the signal quality because you're removing trial level artifacts. And after calculating this difference, the program then extracts the magnitude value at the auditory response frequency bin across all available trials, and then creates a new data set of all of these amplitudes. And then bootstrapping is conducted to generate a distribution of these mean values. And then a 99% confidence interval test is conducted to see if the value of 0 is within the interval. So a value of 0 means here that there was no difference in the amplitudes of the auditory response or auditory response frequency bin when the stimulus was on versus when it was off. And so if 0 is not included, then the program determines that there was a present auditory response. Otherwise, if 0 is included, then the program determines there is no significant response present. The program will continue to collect and analyze data in batches of 10 trials until the 99% confidence interval test passes or the max trial limit is met. And in this test, it is essential to calculate the mean, specifically because it allows us to average out non-auditory related neural background noise that is also electromagnetic noise that is also present in the electrode recordings. And so this is because we're using electrodes that are on the surface of the head. We're not patching any like single neurons. So it's all of these electrodes on the head are also recording all different sorts of neural activity. But luckily, since this noise is random and is not time locked to the stimulus presentation or when I present the sound, if we calculate the mean across multiple trials, the average value of this noise will approach 0, and we can recover the auditory signal that way. But the number of trials needed in the average to recover the response truly depends on the frequency and the amplitude of the sound we're presenting. So for instance, for a signal that is sufficiently high enough amplitude and well within the audible hearing range of the animal, you can calculate the mean across like a dozen trials and observe a clear auditory response. On the other hand, for a stimulus that is low in amplitude and at the edges of the audible frequency range, you may need to average across hundreds or even thousands of trials to see a small blip of a response. And despite this great range of trials that are needed to see a response, most studies use a static trial count for all stimulus types, and we think we can try to save some time there. So by implementing this automated 99% confidence interval criteria in the response detection algorithm, we can achieve adaptive presentation of trials in the experiment online. And compared to a static trial count, this method saves a lot of time and automates much of the test process. And now focusing on the model-based threshold estimation process, since we are measuring neuronal activity, you can generally model its input-output function using a sigmoid. Specifically, a sigmoid effectively models the output of neurons as it relates to input stimulus strength. So the bottom asymptote here represents when the stimulus is at below a detectable level for the neuron or population of neurons, and thus there's no change in output. And then when the stimulus strength is great enough, within a certain range, the neuron is sensitive to changes in the stimulus, and the neuron output will also change. And the upper asymptote represents the physical limit of neuronal output. You know, neurons can only fire so many times for a given period of time. And so the response threshold would be this takeoff point where the neuron just begins to be sensitive to the incoming stimulus. And so in our program, instead of a soft logistic model that I just showed with nice like soft curves, we use a hard logistic, a 3-parameter simple hard logistic model with the auditory threshold value built in as this elbow point parameter here. Another measure of auditory sensitivity we can use is this slope, the slope of the upper arm here. And then the lower arm of the model represents the noise floor in the recordings, so the background neural electromagnetic noise. And a major advantage of this method over many methods used in this field is that the threshold value is estimated using this whole response function. So also accounting for super threshold responses, not just near threshold. And these near threshold data points are most affected by the noise and have the lowest signal to noise ratios. And when I fit this elbow model to previous data sets, we are finding that we can achieve promising fits. And when there's truly no response at any stimulus amplitude, as seen in the 440 hertz condition, you can just model that data as a line. And from these models, we can take the elbow point parameter value at each stimulus frequency we have data for and recreate the audiogram in a fully quantitative way. So, in conclusion, next steps for this work will involve further testing and validation using live animals and directly comparing these results to threshold determination methods that have been used previously. And I hope this work in the future will help other scientists test in a greater variety of species so we can continue learning about the natural world around us and be more confident in our estimations of auditory abilities in these animals. And before I end, I would like to share some quick reflections from my participation in the Accelerator program. And I also took CSC 583 at the same time, and that's computer engineering for data scientists. I really enjoyed the class and recommend it for folks who haven't taken it. And so I'll share insights from that class as well. So first, I finally appreciate how powerful Git and GitHub are. Git is a open source version control software that tracks changes in your files. And GitHub works with Git and hosts your files online and allows collaborators to edit those files as well. And for some people in the room, I know this is like super basic, but it really like changed my life coding. And for instance, I don't need to do this whole code graveyard thing where I like comment out hundreds of lines of code thinking like I might use this, but I never actually do. So my code is just super messy. And then also I don't have to do this like when I change a version of my script, I don't use underscore new, underscore newer, underscore even newer anymore. So I really recommend, it's worth the pain learning how to use these tools. I also learned the value in slowing down when I write code and to take the time to write new code that tests if the other code I just wrote was correct. So these are called unit tests. I've also been spending this past month methodically stepping through the 50 whatever scripts in the package with my hardware online and my electrodes in a potato to do end-to-end tests. And although it takes like a lot of time and progress feels incredibly slow, it reduces the possibility of silent bugs in your code. So your code might run, but it's making mistakes that you aren't necessarily catching. And that's something I'm definitely afraid of. So it's worth doing the checks. And for actual guidance, like step-by-step guidance on how to check your code, I recommend reading Ariel Rokem's paper on this topic called 10 Simple Rules for a Scientific Code Review. And then lastly, I learned that everybody loves histograms. So I learned this from Dr. Brenna Hazelton, who is my professor in CSC 583. Like everybody in research knows about histograms. It's, because we all took similar intro to stats courses. And so histograms can make interpretations of your data more understandable to a wider scientific audience and from different disciplines. So I think, next time you're trying to figure out how to present your data, maybe a histogram would be a good start. And with that, I want to thank my PIs, Joe and Andrew and Ariel Rokem, who helped me so much in this project. And then shout out to my CSE 583 friends and my funding source. And I think I also do scientific illustrations. So if you are interested, I'll take commissions. So yeah, anyways, that's all. Thank you.